Hi, this
Hello.
is Science Hangout from Science Mission. We have today Dr. Chala Eroglu from Duke University with us. She is an assistant professor in the cell biology department at Duke University. She has published several publications, articles in very well-known prestigious journals over last several years. Welcome, Dr. Chala Eroglu, to Science Hangout.
Hello.
Hi. Uh, like, can you please tell us about your background, your education, some of your research interests?
Sure. I am originally from Turkey.
Mhm.
I studied chemical engineering as a
Okay.
bachelor's degree. And
Okay.
after finishing my degree, I decided to switch to um, biology.
Uh-huh.
And I did a master's again
Mm-hmm.
in Turkey in Bilkent University on molecular biology.
Mm-hmm.
Then I went to Heidelberg, Germany, and joined EMBL, European Molecular Biology Laboratories, as a
Uh-huh.
PhD student.
Okay.
There I studied biochemistry of metabotropic glutamate receptors,
Mm-hmm.
particularly from Drosophila.
Okay.
And I did my PhD there. And after my PhD, I realized that I had great interest in understanding the cell-cell interactions in the nervous system, and
Uh-huh.
particularly of those between glial cells and neurons.
Uh-huh.
And to learn about this system and to study this interesting question, I went to Stanford University to the
Okay.
lab of Ben Barres. And there
Mm-hmm.
I started working on the role of astrocytes, the major glial cells in the
Mm-hmm.
brain, on synaptic connectivity. And that experience kind of shaped my research directions here in my own lab. That
Mm-hmm.
I've had my lab since 2008, and we
Mm-hmm.
are interested in understanding how astrocytes and neurons communicate
Mm-hmm.
to form and remodel the nervous system.
Okay.
We are, of course, interested in other cell types in the brain as well, but
Mm-hmm.
predominantly trying to understand this intimate relationship between astrocytes and neurons, and how it's occurring at the neuron-neuron contacts called synapses.
Okay. That is interesting. And what made you change initially from mechanical engineering to biology? It's a big change. So is there somebody influenced you or anybody
Well,
persuaded you?
it was I actually was always very interested in biology,
Mm-hmm.
but I grew up with engineer parents, so
Mm-hmm.
I also had a great interest in mathematics, physics, and so on. So at the point where I was choosing my undergraduate subject,
Mm-hmm.
I was kind of influenced by the fact that I really liked mathematics and physics, and I appreciate the background that engineering training
Uh-huh.
gave me.
Yep.
It's a intense training that is focused on problem solving, and I
Mm-hmm.
think it did help me
Okay.
to form my directions.
Mm-hmm. Mm-hmm.
Um, However, I was always very interested in biology questions. So when
Mm-hmm.
I graduated, I realized I wanted to go after the great unknowns of the biological systems. So,
Yep. Okay. And what motivates you every day? Like, I mean, like there are lots of problems as you mentioned in biology, but what really wakes you up every day and forces you to go to work and do the excellent work what you are doing right now?
well, the fact that we don't know a lot of things and then we can try to figure them out, that's my greatest motivation.
Mm-hmm.
I take great joy in figuring out something that nobody thought about before, and biology research
Mm-hmm.
in general gives us that chance. It's a
Okay.
very creative process, so
Mm-hmm.
there are certain things known, but nothing is really for sure, and you can imagine certain things and go test them. So
Yep.
not only like we can just imagine it, but we can go test it and figure
Yep.
out what's the right thing to do.
Mm-hmm.
So it's really nice. That motivates me, the
Mm-hmm.
intellectually, and also it's a it's great to be always surrounded by really intelligent and motivated people. That's also
Uh-huh.
another thing that motivates me. I like
Okay.
going to work and interacting with all the students, colleagues, and the,
Mm-hmm.
the,
Mm-hmm.
the intelligent people of
Okay.
university. Yes.
And can you please tell us about your lab people, and all those things?
So currently, my lab is located in Duke University Cell Biology Department. I do have 4 postdoctoral fellows
Mm-hmm.
and 1 graduate student with me.
Mm-hmm.
2 other graduate students are rotating to join the lab,
Mm-hmm.
and I also have 4 undergraduates, actually 5 undergraduates that come and
Wow.
work with me. So it's a nice group of people.
Mm-hmm.
It's not too big, it's not too small, just right.
Mm-hmm.
They are all very motivated, self-motivated, and
Mm-hmm.
they are each doing their own interesting research, and
Okay.
that's another motivation for me to go to work every
Okay.
to interact with them. Yeah.
Okay. And what are you going to present today?
I'm going to give you a summary of some of the work we did over the last 5 years.
Okay.
That has showed that astrocytes communicate with neurons of the cortex to
Mm-hmm.
model the certain type of cortical connections that relay sensory information. It's a combination of several articles, but in a way a continuous storyline. So I'm
Okay.
just gonna present that today.
Can you please go ahead and start the presentation,
Sure.
please? Thank you.
Let me know if you are able to see my presentations. Are
Okay.
you able to see my presentation? Okay. Okay.
Yep.
Okay, great. So today I'm going to share with you some of the recent work we've been doing in my laboratory.
Mm-hmm.
We are very interested to understand how synapses form. And
Okay.
for those of you who may not be so familiar with synapses, synapses are essentially cell-cell adhesions between neurons.
Mm-hmm.
So there is a presynaptic neuron which sends an axon, which is labeled in red here, that is laden with synaptic vesicles that contain neurotransmitters. These vesicles are released at synaptic junctions, and on the postsynaptic side, there is another neuronal process. Oops, I don't know how this is so fast moving. Okay, let's go back. Okay, so on the postsynaptic side, there is a um, dendrite from another neuron, and there is a postsynaptic density which has receptors that sense the neurotransmitter release, and therefore the synaptic transmission occurs. So
Okay.
synapses are very important.
Mm-hmm.
We have 10 to the power 14 synapses in our brain estimated, and
Mm-hmm.
they need to be formed in a certain precision, and certain types should be balanced with each other. Otherwise, we would not be able to regulate our cognitive functions.
Mm-hmm.
And almost every, if not all, neurological disorder has some basis in the synaptic a dysfunction in the synaptic component. So it's really fundamentally important to understand how synapses form and how they are regulated and how they function properly.
Mm-hmm.
Therefore, a huge amount of effort in neurobiology have really focused on understanding how these neuron-neuron contacts are formed. However, one thing we more recently noticed that these neuronal contacts, where we call synapse, are not in a void in the
Mm-hmm.
absence of any other structure around them. If you
Okay.
now look at the structure I highlighted in blue, a process coming from a different cell, and that's
Mm-hmm.
an astrocyte process. As you can see, it's right next to the synapse, and
Mm-hmm.
in fact, it's not just a functional interaction; it's a structural interaction. It's also a functional interaction, because functionally these processes have receptors for neurotransmitters. They can detect the neurotransmitter released from one side of the synapse and detected by the other.
Mm-hmm.
And what is also really exciting and remarkable is that it's not that one astrocyte enshoots a synapse. That's just not possible mathematically. In fact, astrocytes are extremely complex cells. Here you see
Mm-hmm.
in blue one astrocyte. And each tiny process of this complex cell would ensheet at least a synapse. And it's estimated a human astrocyte would ensheet around 1 to 2 million synapses at once.
Wow.
So that means it can listen to that many synapses at once, and in
Mm-hmm.
response, these cells do send back signals to the synapse, and those are thought to change the synaptic transmission. That means there is another level of control of synapses at the astrocyte level, which
Mm-hmm.
is something that we're just starting to understand and appreciate. Besides being important for the synaptic function, in recent years, work from my laboratory and others also uncovered that astrocytes are extremely important for the establishment and remodeling of these synaptic structures.
Mm-hmm.
So this is actually the fundamental question that we address in my lab. How do astrocytes control the formation and remodeling of synaptic circuits?
Okay.
So the idea that astrocytes may have anything to do with synapse formation or synapse remodeling ironically came from studies where they have used pure neuronal cultures. So
Mm-hmm.
traditionally, using hippocampal or cortical cultures, neuronal cultures, people have done these know that you have to have your neurons in the presence of astrocytes because astrocytes provide survival factors that keep the neurons alive. However, that eliminates the possibility of too specifically addressing what astrocytes bring to the table. In the lab of Bambaris at Stanford University in the 1990s, early 1990s, they developed methods to isolate pure populations of neurons from the rat retina. These are retinal ganglion cells, as you are seeing on the left corner of the screen here. They are, beautiful neurons. They are all excitatory, and they can be cultured and be really healthy in the complete absence of astrocytes. In a media composition that's totally known, there is no serum here, so we know what's exactly in the media. We can then, culture these neurons in the presence or absence of astrocytes and start asking what do astrocytes do in terms of synaptic connectivity of these neurons. And there is a relatively easy way to determine whether synapses are formed by using light microscopy. So as I showed earlier in electron microscopy, synapses are points where dendrites and axons come together and specialized synaptic proteins come together. So in immunohistochemistry or cytochemistry, if we stain for a presynaptic marker that would be on the axon and a postsynaptic marker that would be on the dendrite, where these two markers appear to localize really close to each other because they are really just at the junction, at the synapse, is what we call a synapse. And of course, we go and verify this by electron microscopy later to really ensure that we're looking at synapses. Okay, when in Barrett's lab, they did these experiments first time, they immediately noticed that, as you see, the retinal ganglion cells here stain for a pre and a postsynaptic marker. The cells we are seeing on the screen are neurons. And you can see on the left, these neurons that were cultured by themselves, they hardly have any colocalized synaptic puncta, whereas the ones that were cultured with astrocytes, which are in the middle, you can see that they have plenty of synaptic puncta. So this led to the appreciation that astrocytes were providing factors that induced formation of synapses. And in fact, you don't necessarily need the astrocytes to be together with the neurons in the same culture dish, but you can simply condition some media with astrocytes and feed this media to the neurons, and that media itself contained the secreted soluble signals that were coming from astrocytes that induced synapse formation. So these these studies showed us that astrocytes secrete signals that induce formation of synapses. And in fact, if you patch onto these neurons, you also realize that they have enhanced synaptic activity that indicates that astrocytes also induce the increase in synaptic function. So this in vitro model proved to be really useful for the identification of these secreted soluble signals. So using this method, many labs, including ours, have now identified a number of really important signals. I'm just going to briefly describe a few of them that will be relevant to our talk today. And interestingly, astrocytes also induce formation of synapses not by just a single shot, single molecule, but by secreting several different molecules and inducing different stages of synaptogenesis. The first stage that they were shown to promote is the formation of a structural or a silent synapse. And by that, what I mean is this synapse structurally looks identical to what I showed you earlier. It has a presynaptic side with the vesicles and a postsynaptic side which has the postsynaptic adapter proteins. However, it lacks AMPO receptors, and therefore, if you patch onto the postsynaptic neuron, you do not detect any increased synaptic function. They do, in fact, have another kind of glutamate receptor, the NMDA receptors, and therefore, these synapses are thought to be the silent synapses. On top of this, astrocytes are also capable of inserting the AMPA receptors into these silent synapses and induce the formation of fully functional synapses. So using this culture system and biochemistry and fractionation of the astrocyte conditioned media, first Barrel's lab and afterwards our lab have identified a number of factors. The first one was identified back in 2005. It is a family of proteins that are secreted by astrocytes called thrombospondins. Thrombospondins induce formation of excitatory synapses. These synapses do not have AMPA receptors in them. They are the silent kind. And in my earlier work, while I was a postdoc, I identified the mechanism through which thrombospondin induces synaptogenesis, and that turned out to be really interesting. I will not go into detail because that's not going to be the topic of our talk today, but it turned out to be a receptor that's also known as calcium channel subunit alpha-2 delta or gabapentin receptor alpha-2 delta. It turns out the thrombospondin binds to these previously unknown receptor and induces formation of excitatory synapses. More recently, the lab of Ben Barres identified glypicans as another family of proteins, glypicans 4 and 6 particularly, which are proteins coming from astrocytes that are capable to insert AMPA receptors on the postsynaptic side, therefore induce the formation of functional synapses. And in my lab, we have identified another synaptogenic protein, and this is going to be the topic of my talk today. We like calling it Heaven, but its official name is SPARC-like 1 or secreted protein acidic rich in cysteine-like 1. Heaven is a secreted protein coming from astrocytes in really high amounts. However, it has no homology to thrombospondins or glypicans, so it's a completely different family of proteins. Heaven in culture induced formation of silent synapses, which resembles thrombospondin, but later I will show you in vivo Heaven's role is very specific and distinct from thrombospondins. And interestingly, astrocytes secrete also a close homologue of Heaven called SPARC. And SPARC and Heaven are homologous. SPARC is not synaptogenic. On the contrary, SPARC is an anti-synaptogenic protein, which specifically inhibits Heaven's synaptogenic activity. So today I'm going to talk about Heaven mostly, but one of the questions, of course, that comes to our mind is why do we have these different signals and what are their particular roles in in the brain to control synaptic connectivity, and how do astrocytes use these different signals to manipulate synaptic connections. So one of the first things we noticed about thrombospondins and heaven, which seem to have the same synaptogenic function, they are actually expressed at distinct time points in the brain development. We like working on mice for our in vivo studies, and in mice, in the mouse cortex, thrombospondin expression is relatively limited to the very early points in the initiation of excitatory synaptogenesis, where this is a time period where dendrites send out these Philopodia scouts that look for appropriate presynaptic partners, and thrombospondin is just upregulated right around time and secreted by immature astrocytes. Whereas heaven is, it starts to be expressed by astrocytes as the astrocytes mature, and its expression actually correlates more with the maturation of these philopodial structures into dendritic spines, which are the sites of excitatory connections, majority of excitatory connections in the cortex. So this showed us that maybe the function of these proteins could be different because they are given by astrocytes at distinct times, and on top of it, heaven expression does not decrease in the adult brain. It seems to stay up and high, so maybe heaven is continuously required to be secreted by astrocytes to also maintain synaptic structures. So another thing that attracted our attention is that right where heaven starts to be expressed is a time point in the mouse development where their sensory experiences start to occur. Mice are born, as you see on the left-hand side, their eyes are closed, their inner ear canals are closed, so they do not receive visual or auditory input until around these P10, postnatal day 10 to postnatal day 15 period. That's when these eyes open and the ear canals open, so the mouse pups start to experience their surrounding. So we also wondered whether heaven protein had anything to do with shaping the circuits in response to sensory experience. So today I'm going to show you two studies that we've done more recently that actually provided us the answer of what heaven was important for in the developing cortical circuits. We realized that heaven is critical for the formation, maturation, and refinement of dendritic spine structures in the cortex. So it correlates with the time that this protein is secreted. And it does this and assemble a specific kind of connection in the cortex. These are the thalamocortical synapses, or the synapses that relay the sensory information that I mentioned just earlier. And we figured out how heaven does this, and it's by physically putting itself in the synaptic cleft between the pre and postsynaptic side and bridging two sides of the synapse. So I will just summarize these findings to you in the rest of my talk. So the way we identified heaven was through a candidate-based screen. We knew that astrocytes made really high amounts of heaven protein and secreted it from gene expression studies and proteomic studies. We decided to test whether heaven could be a synaptogenic protein. To do that, we went back to our retinal ganglion cell cultures and we applied some heaven into these cultures and then determined that heaven addition, as you can see on the picture on the right-hand side, heaven addition to neurons induced the formation of synapses, which are determined by the colocalization of pre- and postsynaptic markers. And heaven protein does not look anything like thrombospondins or any other known synaptogenic proteins. It essentially have two discernible domains. It has an N-terminal acidic domain and a C-terminal domain, which is homologous to another protein that's called SPARC. And SPARC was one of the candidates that we tested when we were trying to identify synaptogenic proteins. But you can see here in the dendrite stretches, I cannot move the mouse. Oh, yeah, I'm able to. I don't know if you can see my mouse, but here when we treat the RGCs with Spark, we do not see colocalized synaptic puncta, indicating Spark is not synaptogenic. However, what was surprising was that Spark was not synaptogenic, and in fact, it was anti-synaptogenic, because when we treated the neurons with a combination of Heaven and Spark together, Heaven no longer was able to induce synaptogenesis. So here's a quantification of that. Heaven increases the number of synapses significantly from when the neurons are cultured by themselves, but Spark is capable of inhibiting that. And Spark is specifically inhibiting Heaven, it's not inhibiting thrombospondins. So this told us that Heaven was inducing synaptogenesis probably through a mechanism that was also distinct from thrombospondin-induced synaptogenesis. Another thing we've done in this earlier study in 2011 was to investigate how the synaptic connectivity was altered in the brains of Heaven and Spark knockout mice. We looked at a particular part of the brain, which is called superior colliculus. That's the part of the brain where retinal ganglion cells, the cells that we culture in vitro, make their normal connections in vivo. And when we went and analyzed the connectivity at this part of the brain for Heaven knockouts or Spark knockouts, we found that Heaven knockouts, in line with in agreement with the role for this protein in synaptogenesis, had diminished synapse numbers, whereas the Spark knockouts had an increased synapse number. It's showing that Spark is an anti-synaptogenic protein. So these results were exciting, and we really wanted to understand more about Heaven and Spark and study this in further detail. And we also did some electron microscopy, because this is a very nice method to really visualize synapses, which are sub-micron structures. And when we did electron microscopy in the Heaven knockout knockout or wild-type mice at 25 days old, here I'm going to highlight, you can see that on the postsynaptic side, the postsynaptic densities were significantly diminished in the Heaven knockouts here on the right. Whereas on the presynaptic side, also the bouton size and the number of vesicles were greatly diminished in the Heaven knockouts. So this told us that Heaven was not only important for the formation of synapses, but its lack also caused immaturity or morphological defects in the mice. But one thing we didn't immediately notice from these two-dimensional representation of the synapses was that also the location of the synapses appeared to be different. And this was noticed by a really talented postdoctoral fellow in my lab, Chris Risher. Chris, when he started looking at these pictures, he realized that Heaven synapses were made directly and onto dendritic shafts, whereas the normal synapses are usually made directly onto a dendritic spine head. And I'm going to illustrate this further in the next slide. So it's hard to visualize this in 2 dimensions. However, there is a method where you can build 3-dimensional reconstructions of dendritic stretches using serial section EM. And that's something that Chris did. And when he did this for a wild-type dendrite, for example, here the example, you can see this dendrite has nice spines that are coming out from it. These spines have a thin neck and a nice bulbous head. And this is what you would expect in a wild-type situation. However, when he looked at Heaven knockouts, instead of nice spines, they still have immature-looking philopodia that are coming out from the dendrites that didn't look anything like the wild type. And furthermore, when you look at where the synapses are at the tips of the dendritic spines normally, whereas in Heaven knockouts, they were mostly on the dendritic shaft. So this already gave us an idea that heaven might be really critically important for the formation of these dendritic spine synapses. However, dendritic spine synapse formation, or the timeline of its development, is not really well characterized in the superior colliculus, which is where we've been studying this question. Therefore, Chris decided to work on a different area of the brain, which is the visual cortex. This is a part of the cortex where visual inputs that are relayed through thalamus are processed, and it's marked here where it would be on a mouse brain. And in this part of the brain, studies from decades have shown us that the dendritic and the particular region he's focusing on is the V1. And in this region, it's well known that early in development, dendritic spines start as filopodia, and as they form synapses, they slowly mature into, okay, let's go back there. They slowly mature into these more mushroom-type synapses. So he wanted to determine whether heaven knockouts had any problems in this maturation process, and he first used a very old technique, the Golgi-Cox staining, that was used by many scientists over the last century. And he did in fact found that heaven knockouts did indeed had more immature looking filopodia. This gave him the courage to do the more painstaking analysis by three-dimensional EM, and he could verify that heaven knockouts did indeed in the cortex had these more immature looking spines. However, as you can notice, this is not a phenotype that is as strong as the one we observed in the superior colliculus. And another thing that was different from that part of the brain was that heaven knockouts in the cortex, even though the synapses were immature, they did form equal number of synapses. So one thing, of course, we could take away from here is that heaven may not be required for the formation of cortical synapses, but it is important for the maturation of cortical synapses. However, we then realized that was not the case. It was the following that made us realize maybe there's something more complex going on here. So in the cortex, there are two classes of main classes of excitatory synapses. And the first class, which is very abundant, where we are studying synaptogenesis, is the intracortical connections. These are connections between cortical neurons made by other cortical neurons. And luckily, there is a marker, a presynaptic axonal marker that we can use, which is a vesicular glutamate transporter 1. And that is a way to mark these intracortical synapses. But there is another population, which are the synapses that come from the thalamus, which are positive for another protein called VGLUT2. So these thalamocortical synapses are fewer in this synaptic zone where we study synaptogenesis. However, they still constitute a reasonable amount of the synapses there. So Chris wondered whether heaven specifically affected the formation of any of these classes of synapses. And he found the following. So in heaven knockouts, unlike what we've seen before in the superior colliculus, the intracortical synapses were in fact actually higher in number. But that increase in intracortical connectivity was in expense of a dramatic decrease in the thalamocortical synapses. So this made us think that maybe heaven was specifically inducing formation of thalamocortical synapses, which are VGLUT2 positive. But in the absence of heaven, the lack of those synapses that did not form were compensated by the formation of more intracortical synapses. So we were seeing, therefore, at EM level, where we cannot differentiate which type of synapse we have, except the fact that they are excitatory, that we were seeing equal numbers of synapses forming. So to further verify this, Chris did the following experiment. He injected purified heaven into the brains of heaven knockout mice, and really remarkably, within 3 days, this induced formation of VGLUT2 positive synapses. So that showed us that heaven could act locally right at the cortex to increase these thalamocortical synapses. And here's a quantification of that. This also, came together very well with another observation that we made in our electron microscopical studies. So quite universally, it's believed that when you see a spine on a dendrite is a site for a single excitatory synapse. However, in heaven knockouts, Chris very often found these spines which contain 2 excitatory synapses made onto them. And these he called the spines with multiple excitatory contacts, or SMACs. These SMACs could come in several different configurations. The most common ones were the ones where both contacts were made by distinct axons but were made onto the same spine head, or one was at the spine head and the other was at the neck. So this was quite interesting. This showed us that spines may actually be sites for multiple innervation under certain conditions. And when he quantified this for wild type or heaven knockout mice, he found that SMACs were very prevalent in heaven knockouts, but not so much in the wild types. It was pretty rare in the wild types. Because heaven knockouts had so many immaturity problems, one thing we hypothesized was that maybe these structures occurred in wild types as well, but were at an earlier developmental stage. And to test that, he did the reconstructions in P14 wild type mice brains, and he indeed found actually quite a considerable portion of the excitatory synapses. Around 25% of all excitatory synapses are made onto these structures. So this made us to hypothesize that these were structures where potentially different axons competed for sites of interaction with dendrites. And because we knew that heaven was critical for thalamic connectivity, we wondered whether there was anything with the axons. This is a movie, I don't know if it's going to play in here, but didn't necessarily play. But when he went and reconstructed the axon, he found that if an axon started with a multiply innervated SMAC, and if you followed that axon, its likelihood of hitting another SMAC, which hopefully we'll show in a second, there you go. So when you look for what other synapses these axons make, their likelihood of making another connection with another double innervated spine was astronomical. It was 100-fold higher than log. So that means there's something special about these axons. They're either more aggressive and going and taking over the synapses from other axons, or they are weaker and they are losing their connections to other axons. We don't exactly know that yet. However, since we know heaven is critical for thalamic connectivity, Chris wondered, could these SMACs be sites for competition between thalamic and cortical axons? And to test that, he did immunogold labeling to label for markers of VGLUT1 or VGLUT2 synapses, and he could visualize one SMAC where one side of the SMAC connected to a VGLUT1 synapse, a cortical synapse, and one side connected to a thalamic synapse. So, of course, this is still ongoing work that we really want to figure this out. However, it seems like these multiply innervated structures are structures where thalamic and cortical axons potentially either compete or collaborate to form these synapses. So our model right now is that initially in development, spines may serve as sites of competition between distinct inputs, and astrocytes, by providing heaven, stabilize the VGLUT2 synapses, and therefore the spines that where VGLUT2 would win the competition, that competition is won. And we don't know exactly what are the ways that these synapses are stabilized fully yet. That's something that's ongoing work. But when we have a situation where heaven is not there, what we think is happening is that VGLUT2 synapses cannot fully stabilize, and therefore they are still taken over by the intercortical VGLUT1 positive synapses. So, so far I've told you what heaven coming from astrocytes is doing in the cortex, mammalian cortex. But of course, you might be wondering, how is heaven doing it? And that's the rest of my talk. That is the molecular mechanism through which heaven induces formation of thalamocortical synapses. And this is work by another very talented postdoc in my lab, Sandeep. Sandeep asked the question, what is the molecular mechanism of heaven-induced synaptogenesis? And when he started this work, he knew already that heaven is a protein that is physically localized to synapses. So that gave us one clue. Even though heaven is made by astrocytes, it is at the synapse. And from our electron microscopy work, we know that heaven controls formation of both pre- and postsynaptic structures and their maturation. Therefore, we hypothesized maybe heaven is a synaptic linker protein. And what do I mean with synaptic linker? Let me just briefly describe it. So there are neuronal synaptic adhesion proteins that bridge the pre- and postsynaptic specializations and recruit pre- and postsynaptic machinery. What we thought heaven might be doing is that heaven secreted by astrocytes further bridges these specializations to each other and therefore recruit more of the pre- and postsynaptic structures and organize the formation of a larger, more developed, more organized structure with more of its components. So if this hypothesis is true, then we expected that heaven should at least have one pre- and one postsynaptic receptor, and it should have the capability of interacting with both of these receptors and linking them across the synapse. Through several different methods, Sandeep identified the pre- and postsynaptic heaven receptors to be none other than the neurexin-1 alpha and neuroligin-1. I'm not going to go into detail how we identified those. You could find the details in the paper. However, what's interesting is when you do an immunoprecipitation with heaven, you can immunoprecipitate both neurexin-1 alpha, which is a presynaptic protein, and neuroligin-1. And interestingly, just to remind you, heaven had these two distinct domains. And if you just use the C-terminal end of heaven, that still, to an extent, can interact with neuroligin-1, but it cannot interact with neurexin. So you need the full protein to be able to interact with neurexin-1 alpha. Using biochemistry, Sandeep identified the part of heaven that is absolutely crucial for interaction with neurexin and neuroligin and for induction of synaptogenesis to be at the cusp of these two domains. We call it the DE domain, but it's just an arbitrary name. What he found that neurexin-1 alpha and neuroligin-1 have binding sites right around that region, and they do bind to heaven through those regions. What's interesting about neurexins and neuroligins are they are well known to be important for synapse formation and synaptic maturation and function, and they are known to interact with each other. However, there is one important detail here. The neurexins come in at two different flavors, and the flavor that people have studied mostly is the neurexin betas, which I will come to that, and those are the ones that strongly interact with neuroligin. Before I go there, I'm just going to briefly show you an experiment Sandeep did. He made a heaven protein by that a mutant by deleting this region. So if he deletes this region critical for interaction with these two receptors, that protein no longer is synaptogenesis, inducing synaptogenesis. Whereas full-length heaven is fully capable of inducing synaptogenesis, or if you delete another portion of heaven on this bit here over at the N-terminal, and that doesn't impair its function. So this is really the critical functional domain. So we thought that we knew that heaven did interact with 1 alpha, but did not interact with the shorter Norexin 1 beta. Norexin 1 beta is capable of itself interacting with neuroligin 1. However, when we looked at the literature carefully, we realized that Norexin 1 alpha and neuroligin 1 do not interact with each other. Therefore, we hypothesized maybe that's what heaven does. It is able to bridge these two otherwise uninteracting neurexins and neuroligands across the synapse. And how to test that? Sandeep used a method that was developed by Peter Scheifele and Anne-Marie Craig's labs. It's really a neat cell biological method where you express neurexins in a non-neuronal cell. Here we use Kosovar hex cells, and you put them together with neurons that are expressing neuroligin 1. And if neurexins that you express, for example, it's a neurexin beta, it would induce neuroligin 1 to interact with it. But if it is neurexin 1 alpha, which doesn't attract neuroligin 1 to its then to these non-neuronal cells. Now our question is, can we take this situation where we have the heaven receptors which don't interact with each other, or we call them incompatible, and add heaven and turn that into an interaction? So here are our positive and negative controls. Again, neurexin 1 beta can interact with neuroligin 1. So you see nice neurites contacting this non-neuronal cell, whereas neurexin 1 alpha doesn't interact with neuroligin 1, so no interactions. Now we add heaven to this no interaction situation, and there you can see heaven now induces interaction between these two otherwise uninteracting receptors. And remember the mutation that rendered heaven incapable of inducing synaptogenesis. If we use that mutant protein, we no longer induce the bridging between these two receptors. So these findings told us that heaven's bridging of neurexin and neuroligin was critical for its synaptogenic function. But we wanted to further understand what this bridging meant for in vivo mouse-brain connectivity. And for that, we went to the cortex, the same region that I showed earlier. We did the earlier work with electron microscopy. And just to remind you again, this region contains synapses from cortical axons as well as the thalamic axons. And heaven knockouts, because heaven is critical for the formation of thalamocortical circuits, heaven knockouts have severely decreased thalamocortical connections, which are VGLUT2 positive, but they have an increase in their VGLUT1 positive intracortical connections. So a very talented graduate student, Jeff Stocksdale, collaborated with Sandeep on these experiments, and he went and studied the connectivity in heaven receptor in the knockouts of heaven's receptors, the neuroligin and neurexin knockout mice. And he found that they had a very similar phenotype when compared to heaven. So they did have increased connectivity between intracortical neurons, but they did, both the neurexin and neuroligin knockouts had that, whereas they had diminished connectivity from the thalamus to the cortex, in line with the fact that these are heaven receptors. And to test whether bridging of neurexin and neuroligin was critical for the formation of thalamocortical synapses, Jeff used the method Chris had used before. Remember, we can inject heaven into the heaven knockout's brain and induce formation of thalamocortical synapses. Jeff repeated this experiment, but he also injected in some of the mice the heaven mutant, which is not capable of bridging orexin and neuroligin. And that in itself was incapable of inducing thalamocortical synaptogenesis in vivo, further showing us that this bridging of orexin and neuroligin is critical for the formation of thalamocortical synapses. So these findings together showed us that having coming from astrocytes assembles formation of thalamocortical synapses by linking orexin 1 alpha, which is presynaptic, to neuroligin 1. But what does this mean in terms of the function of the cortex, especially the visual cortex where we're studying, and in terms of the ability of the cortex to modify itself, cortical connections to modify themselves in response to experiences? To test this, we collaborated with Alex Medina's lab, Nisha Pulimode, and others in his lab to determine if heaven was involved in visual circuit function and plasticity. And to do that, they used a paradigm, a plasticity paradigm, well-established paradigm called ocular dominance plasticity. In really simple terms, this is a plasticity that occurs only during a limited time during development. That if you close and if you, for example, close one eye during this period starting around the 26 days after birth, and if you close the eye, oops, how did that happen? Let's go back. Okay. If you close one eye, then what happens is that the other eye, which is the weaker eye, labeled IPSI here, that starts to gain more strength and wins over territory of the closed eye. So normally the contralateral eye has stronger connections to the cortex than the ipsilateral eye. But by closing the contra eye, you can equalize the connections between both eyes, only in this critical period. So Alex and his colleagues made this experiment in wild type and compared it to having knockout mice. And in wild type mice, you would see that that before the eye is closed, the bias, which is basically the ratio between the responses to the contralateral eye versus the ipsilateral eye, are above 1 because the contra eye is stronger. But after eye closure for that period of time, 10 days, this bias is diminished, and now the ratio between the contra and ipsi is more close to 1. This shows that the wild type has ocular dominance plasticity. However, having knockouts, there were 2 phenotypes. First, they were more biased towards the contralateral eye, more strongly connected, and they did not switch their bias to the ipsilateral eye by closure, so they were defective in ocular dominance plasticity. So this told us that heaven is important for this to occur, but we wanted to also know whether heaven coming from astrocytes was driving this, and it wasn't something with the knockouts in general that was wrong, that occluded the plasticity. And to test this, Sandeep and Jeff made viruses that would express heaven back in astrocytes, and they've injected these viruses to the heaven knockout brains. And you can see here an astrocyte labeled in green, and this astrocyte is in a heaven knockout, so normally it should not be stained for heaven protein. However, because we injected this virus that makes heaven, now this astrocyte is making heaven. And when we do this treatment to the heaven knockout mice, we could now rescue both phenotypes that we've previously seen in heaven knockouts. They do have a normal contralateral bias before the manipulation, and after monocular deprivation, they do exhibit plasticity. So taken together, these findings show that astrocyte-secreted heaven is required and sufficient for thalamocortical synaptic plasticity. So in summary, I would like to tell you that I've showed you today astrocytes control circuit-specific, in this case, thalamocortical circuit-specific synapse formation, maturation, and refinement by secreting heavin. And heavin assembles thalamocortical synapses by bridging neurexin 1 alpha and neuroligin 1. And it is in the work that I didn't show you, but is present in our paper. We've seen that this bridging is critical to bring in certain complements of the synapse to the synapse, and these include neuroligin 1, NMDA receptors, and postsynaptic density proteins like PST95 and Homer. And it seems like this organization at the synapse is something that is critical for having plasticity in the cortex. This is also interesting to us in the sense that recent studies, here is a picture from a recent paper in Nature that have sequenced the genomes of a large number of autism patients and their families, and have identified de novo mutations in many genes. And very interestingly, they realized that the mutations were in genes that seem to cluster around synapses. And I really like the fact that they put here neurexin 1 and neuroligin 1, and in an appearance that they interact with each other. But if you remember from my earlier slides, that these two receptors don't actually interact with each other. They do indeed need heavin. When I saw this picture, I said maybe this data that is set would also include heavin, and indeed it does include heavin, or spark-like 1, which is the gene name for this protein, and puts it in fact in the middle of these autism risk genes. So one of the things that is interesting for us that maybe this organization, which is critical for thalamocortical synapse formation and plasticity, is critically important for the function of the brain, and therefore if there are genetic or other errors that occur in the synaptic system, it may be involved in the pathology of diseases, including autism. So I would like to end my talk by thanking the individuals who did the work. Of course, many people in my lab had contributed to this work, but these work were initiated and carried on by Chris Risher, Sandeep Singh, and Jeff Stockstill. I also thank to my collaborators for all their help and the resources that they provided, and for reagents to generous other scientists who shared them with us. And of course, I would like to thank the funding sources that helped us to carry out this research. Especially this research was funded by an NIDA ARA1 to my laboratory. And thank you very much for your attention and listening to me.
Is it possible to? Okay, good.
Can you still see me, or?
No, it is just the presentation. Okay. So thank you very much. It's a very nice presentation. I looked at the cell paper, but I didn't have a chance to go through it. But your talk gave us a really comprehensive idea on like what is happening.
Okay.
I had one question about the whole thing. Like you had initially mentioned about Spark, which antagonizes the interaction, or like which prevents Homer from acting. So how does that play a role in here? Have you done any double knockout mouse or anything like that?
Yeah, those studies are ongoing.
Okay.
Our initial simplistic model was that Spark would block heavin-induced synaptogenesis by potentially just binding to one of the receptors and not binding to the other, and therefore break the linkage. That didn't turn out to be the case. There is a bit more complex regulation here, which is something that we're following through. And besides Spark, there may be other ways that heavin is regulated as well. The two domains of heavin, where they come together, is very critical for its function. But right where the critical domain is, there is a protease cleavage site, and it's cleaved by certain brain proteases. And it makes two two parts of heaven protein which are not synaptogenic anymore, but they may
Okay.
indeed themselves have functions, and which is something that we're testing right now.
Okay, so it's pretty interesting. So really, I wish you good luck in your future work and future research. And thank you very much for taking time to do this science hangout with us. I really appreciate your time and effort.
Thank you very much.
And
It's nice. Thank you for contacting us and giving us the opportunity to share it.
and do you think, like, if you have any, like, if you think that you want to share with more research down the road, few months down the road, certainly you can contact us. I really appreciate you.
Okay, sounds good, Sadashwa.
Thank you.
Take care. Bye.